Hello ladies and gentlemen, welcome to my YouTube channel. I managed to find a typical exam question in Combined Science paper 3 that was on food taste. It was examined in June 2019 and total number of marks on that question was 20 marks. It is very important, guys, that you pay attention to colors observed when carrying out the practical and also be able to interpret nutrients present. Yesterday I uploaded video on food taste. Make sure that you understand the meaning of those color changes. The practical need to be carried out in the lab. Unfortunately, I don't have access to lab, but I will use the marking scheme for that question. This will help you to have a better understanding on practical carried on food tastes. So instruction to candidates, they are required to record all experimental results and show the essential steps in any calculations in the spaces provided in the question paper. Number 1 is reading. You are required to identify nutrients present in juice X. You are provided with 3 boiling tubes, a test tube rack, a pair of tongs, a graduated syringe, Benedictine solution, hot water bath, juice X, solution P, solution R, and access to a clock. Carry out the tests described in table 1.1 below and record the observations and deductions in the table. I will show you the images of laboratory equipments that were used in that practical exam so that you have a better understanding. Candidates were provided with 3 boiling tubes. A boiling tube is a small cylindrical device that is used to rapidly heat items in Bunsen burner. The difference between boiling tube and test tube is that boiling tube is wider. Candidates were also provided with a test tube rack. It is used to hold upright multiple test tubes at the same time. Candidates were also provided with a pair of tongs. They are used to grip something and lift it. So in this exam, it was used to lift boiling tubes. You can't hold it with your hands because it will be hot. Candidates were also provided with graduated syringe. It is used for the measurement of small volumes. In this exam, it was used to measure small volumes of juice X, solution P, and Benedictine solution. Benedictine solution was also used in this practical. It is a combination of sodium carbonate, sodium citrate, and copper sulfate pentahydrate. It is used to detect the presence of reducing sugars. Hot water bath was also provided. The role of hot water bath is to incubate samples at a constant temperature over a certain period of time. They were also provided with juice X. It was orange juice directly from orange fruits. Solution R was also provided in the exam. It was iodine solution. Iodine reacts with starch due to the process that is called complexation. Iodine slip into coils of starch molecules, forming stable complex that results in blue-black color being formed. And then finally, they were also provided with a clock or a stopwatch, which is a device that is used when time periods need to be measured precisely. Let us now go straight into the practical. After carrying out the test, candidates were required to record the observation and write the conclusion in table 1.1. This carried out a total of 10 marks. The first step was to add about 2 cubic centimeters of juice X into a boiling tube and add 2 drops of solution ARA to the juice. So we are going to use a graduated syringe to measure 2 cubic centimeters of juice X into a boiling tube, and then we are going to use a pipette to take 2 drops of solution ARA and add to juice X. So according to the marking scheme, it was converted from brown to blue black. That is the observation that was observed. So it implies that starch was present, because when starch is present, we are going to have our iodine solution, which is solution ARA, being brown in color. It is going to be converted to a blue-black color, showing the presence of starch. So this is what you record. Solution ARA changed from brown to blue-black. This shows that starch was present. And then in the next step, we are supposed to place about 2 cubic centimeters of juice X into a clean boiling tube and add about 2 cubic centimeters of Benedictine solution to the juice, and then we heat the mixture in a water bath. So you should know that Benedictine solution is blue in color. So when reducing sugar is present, it is going to be converted from blue to brick red. So according to the marking scheme I have, it is saying that the observation was a brick red color. So this implies that reducing sugar was present. If it was absent, it was going to remain blue. And then in the third step, place about 3 cubic centimeters of juice X into a clean boiling tube and add about 4 cubic centimeters of solution P to the juice. Place the test tube in the hot water bath and leave it for about 3 minutes. After 3 minutes, pour about half of the contents into another clean boiling tube and then add 2 drops of solution ARA to one of the portions. So according to the results here, it is shown that it remains brown. The solution ARA, which is iodine solution, remained brown. So this implies that starch was absent. And then finally, add about 2 cubic centimeters of Benedictine solution to the other portion and heat in the water bath. So according to the answers here, it is shown that Benedictine solution changed from blue to green. So this implies that there were traces of reducing sugar. Little reducing sugar was present. So this is how you obtain the first 10 marks on number 1A. Let us move on to 1B. On part 2, we need to identify the solution ARA. Solution ARA is iodine solution because iodine solution is the one that is brown in color. When starch is present, it is going to be converted from brown to blue-black. So solution ARA is iodine solution. And then on part 3, we are supposed to state the color of Benedictine solution. It is blue in color. I think I have shown you the image of Benedictine solution. It is blue in color due to copper sulfate solution. That is one of the components that is used in the manufacture of Benedictine solution. And then on part 4, we are supposed to state the nutrients contained in juice X. So we are having starch and reducing sugar because iodine solution was converted from brown to blue-black, indicating the presence of starch. And also the Benedictine solution changed from blue to brick red, showing the presence of reducing sugar. And then on part V, suggest one advantage of drinking juice X. We can see that in juice X we are having starch, we are also having reducing sugars, so it means that it is going to provide us with energy. And then on V1, which is part 5, part 6, suggest a possible identity of solution P. Solution P might be an amylase enzyme. And then on part 7, we are supposed to state the process responsible for the change that occurred when solution P was added to the juice and the mixture heated. So before I attempt this question, let me explain why I suggested that amylase enzyme might be the solution P. It is because previously we tested and we find starch present, and then after adding solution P, we didn't find starch. We have a negative test result for starch, so it means that starch was converted to a reducing sugar by an amylase enzyme. So that is the reason why we are saying P is amylase enzyme. And then on part 7, the process that is responsible for the change that occurred when solution P was added to the juice is known as digestion. If we digest starch using amylase enzyme, we are going to have a reducing sugar. And then on part 8, we are supposed to name the part of the alimentary canal where the process mentioned in part 7 occurs. Digestion, it occurs in the mouth, it occurs in the duodenum, it occurs in the ileum or small intestines. And then finally on part B, we are supposed to state N2 precautions that should be taken when carried out the experiment. We need to wear protective clothes, we need to use the gloves and also avoid spillage. We also need to use the tongs to handle hot test tubes. And also we need to handle hot water with care. That was all about food taste. That was in Combined Science paper 3 in June 2019. Thank you so much guys for following me on this channel. Please don't forget to subscribe, like and share my videos. I love you all. This is Eve signing out.